I'm Jo Chamberlain, the Executive Director of the Coastside Land Trust, and it's really a pleasure to welcome you today to our Ecology of Soil, or Soil Ecology, that will be presented by Dr. Chelsea Carey. And before we get into our presentation, a little bit of logistics. This webinar is being recorded. It will be on our website, and if you are a registered participant, you will receive a link to this recording at the end of the program. And please don't hesitate to share it with anyone you think could benefit from this information. Kate will come on after Chelsea's presentation to walk us through Q&A. If you have any questions during the presentation, at the bottom of your screen, there's a dock, and it has a Q&A button, and you poke that and type in your question, and then Kate will work with you and Chelsea to make sure we get these questions answered. If we don't have time to get through all the questions, we often do a follow-up email clarifying any information that you need or that we can help you with. Let's see. As always, these are free. However, if you would include us in your annual giving, we would be very grateful for your support so that we can continue to bring you these wonderful programs. Today, we are welcoming Chelsea Carey to present on soil ecology. Soil ecology is a fascinating science, and she is on the leading edge of this miraculous research that's being done. Who knew that we're walking on a living earth, and just fascinating the number of species and the sophistication of the living earth, literally the living earth that we are on. Chelsea is from the Midwest, where she started her education in Chicago. Fortunately for us, she came to California and started participating in the UC system, where she was able to complete her PhD at Merced. And we are so fortunate to have her with us. Some fun things about Chelsea: she has a 4-year-old son, and she's a significant marathon runner, including the Big Sur Marathon. So not only is she brilliant, she's an incredible athlete and a mom. So Chelsea, welcome, and let's hear about your presentation.
Thank you so much, Jo, and thanks for the invitation to spend Saturday morning with you all, and thanks to everybody for joining and participating in this event. Okay, so I'm just going to take a moment and share my screen so I can get my slideshow up. Let's see. I think this is what worked last time. And Jo, can you confirm that you can see my screen okay?
It looks wonderful, and incredible little creature, and teeth that are just amazing.
Perfect. Yes. So this is one of the fun things that we're going to be able to do today, is walk through a lot of content and illustrations from what we call the microbe guide, or a guide on the life below ground on the range. And so this is an illustration from Mel Preston, one of my former colleagues, and I'll do a little bit more introduction to that in a moment. But thank you again for having me here, for the invitation. Thanks to everybody for joining. My name is Chelsea Carey. I serve as the Director of Soil Research and Conservation at Point Blue Conservation Science. We are a nonprofit based out of Petaluma, California, so just north of San Francisco, and we're focused on climate smart conservation. So what that means is that we have about 160 staff who collectively work to achieve to build in adaptation and resilience into our landscapes, and where possible, help to figure out how we can mitigate climate change as well. So we see a lot of opportunity to achieve our goals on working lands by partnering with farmers and ranchers. And I've had the pleasure of helping to lead our soils program over the past 6 or 7 years now, helping us to better understand the soil ecology that underpins rangelands in particular across California, and help to figure out and how we can steward these landscapes from a below ground perspective for all of the sort of outcomes and things that we collectively care about. I have the good fortune and pleasure of working closely with partners at TomKat Ranch, so I am down that way frequently, and also get to work with ranchers across California through other programs we have at Point Blue. So really looking forward to talking about the ecology of soil with you all today. I have my PhD in soil microbial ecology and biogeochemistry, so nutrient cycling, carbon cycling, and really looking at the organisms below ground and trying to understand them, who drive all of those functions. The ecology, so if we think we're talking about the ecology of soil, thought it might be helpful to just sort of name what we mean by ecology. And ecology is the study of how organisms interact with each other in their environment. And so, as Joe mentioned, below ground world is living and breathing. It's this miraculous world below our feet that often we aren't aware of or don't think about. And so we're going to unpack some of that today, and hopefully sort of inspire wonder and awe related to what is going on below our feet. So this presentation draws from much from the Life Below Ground on the Range guide that I had the pleasure of writing a number of years ago in collaboration with Mel Preston, who is a former colleague of mine. She's an amazing artist and has produced most of the illustrations that I'm going to share today throughout the presentation. So hope you get to appreciate those. And if you're interested in seeing the guide itself, we have both an online version that you can find at this link that I posted here, but we also have a lot of hard copies. And I would be happy to, if you want to follow up afterwards and you're interested in having some hard copies, to send them your way. So I thought we could begin by talking about why we might or should care about the life below our feet. And then from there, move on to better understand and talk about what influences the life below our feet. So both the presence and activity of soil organisms, and thus all of the services and functions that they provide. And then we'll go in and sort of discuss some important groups of organisms below ground, just a little bit, just a little teaser about earthworms and nematodes, protozoa, fungi, bacteria, and then I'll sort of walk through a couple other notable groups of organisms that we won't have a lot of time to talk about, but that are worth mentioning. And close us out by thinking through how we can steward the life below ground now that we've had a chance at the end to think about why we care, what influences them, who they are, and wrap up by thinking through then more explicitly, how can we promote life below ground? So why should we care? Why should we care about what's going on below our feet? Well, I think an easy place to start is by appreciating the sheer abundance and diversity of organisms who inhabit the soil. So one of my favorite sort of factoids is that in a handful of soil, there are more organisms than there are people on the entire planet. That's an incredible amount of organisms. It's billions of organisms. And with that abundance comes also an incredible amount of diversity in kinds of organisms within that handful. So next time, if you're gardening later today, and you have that opportunity to just pick up a handful of rich soil, I hope you think about that, just that there is a life in that soil that mirrors and exceeds the life we have sort of above ground across the globe. And in on the left-hand side of this slide, I thought I'd just put a table to show, sort of illustrate some of the diversity that resides below ground. So this is a table that looks at different groups of organisms, from plants all the way through then lots of different kinds of soil organisms. It characterizes or lets us know how many known species there are, how many species we estimate to be within that group, and then gives us a sort of percentage of described. So that would be the how many known species you have divided by the total number of species we think there is. And there's lots of different ways to estimate that total number we think there is. But let's just look at the vascular plants. So above ground, there's about 350,000 known species of vascular plants. And we think that that's about 88% of all species of plants that exist. So pretty well characterized. If you move down and let's just look at, nematodes. We've described somewhere between 20 and 25,000 species of nematodes, and we anticipate there's somewhere between 1 million and 10 million species. So with that estimate, we've only scratched the surface on upwards of 2% of the species that have been described. But nematodes are thought to be the most abundant animal on the planet. And then that sort of the magnitude holds when you look through other different groups of soil organisms through, let's say, bacteria at the bottom of this table. We've described about 15,000 species, but we think that there's more than 1 million. So we've characterized really only less than 2% of bacteria. So just this incredible amount of diversity that is really the last frontier for us to discover from a biodiversity conservation standpoint, who they are, what they're doing, but we know that that they're just providing so many services for us. And I thought it would be nice to just contrast this table with which has a lot of numbers with a really beautiful picture on the right hand side. So this is a picture of agar plates that grow different types of bacteria. In this case, it's actinomycetes. So they're bacteria that can be filamentous and grow similar to fungi, and they do a lot of decomposition work in the soil. So they help to break down plant material. So the different colors and the different agar plates are just characteristic. They illustrate different species. And so even just within a single type of bacteria, you just get an incredible amount of diversity. So all of these organisms, so there's so many, there's an incredible amount of diversity, and they're all doing really important things. And we have, I think, just scratched the surface of sort of understanding who does what, but generally we understand that this below ground world is driving so many of the things that we rely on as humans and as a civilization. These organisms below ground, they drive carbon cycling, they drive nutrient cycling, they help to cycle water. So if we, I would argue that even if folks don't know it, and many probably don't know it, everybody cares, not only just about soils, but about the organisms within the soil ecology. Everybody cares about soil ecology, even if they don't know it. And that's because of the far-reaching impact of the organisms below ground. They influence soil fertility and forage production, crop production. They influence the quality of our food and of our feed. They influence plant composition. So whether or not a pasture has, invasive weedy plants overtaking, or is comprised of rich native biodiversity. The below ground world influences water storage and quality. They influence carbon sequestration and the climate regulation. They influence air quality. So all of these, just incredible ecosystem services are driven by the life below ground. And I like to think about it, the analogy, the way I think about it in my mind is that the above ground world is comparable to a Broadway show, right? That's what we see. We see the main stage actors, we see the performance, but a Broadway show is not possible without the backstage crew. And the organisms below ground are the backstage crew. What we see above ground and life as we know it would not be possible without them, even if we don't see them on a day-to-day basis. And this is one of those beautiful illustrations from the guide on the right-hand side that I mentioned. This is from Mel Preston, and it can be found in our microbe guide. And it's illustrating 2 species of dung beetles. And dung beetles are just are a type of organism that helps to break down. One of the services that they provide is that they help to break down both break down cow pies. They help to break down dung from native grazers, cows, and everything in between. And these are 2 illustrations of 2 dung beetles that were introduced actually to California. So on the top, you have the European dung beetle, and then on the bottom, you have a male and female of a bullheaded dung beetle. And they were introduced in around the 70s by actually UCANR, so the UC system, to help with pest fly control and nutrient cycling on California rangelands. So recognizing even back then the importance of these soil organisms for soil fertility and also pest control. So that's just one example, and we'll go through many examples here in a minute through looking at some of these organisms more closely, but just one example of the important role that dung beetles in particular can play on rangelands. And then again, we'll talk more about this in a minute as well, but also just highlighting another quick example of the importance of the life below our feet. Mycorrhizal fungi are fungi that form close associations with plants. The vast majority of plants across the globe have associations with mycorrhizal fungi. These are fungi that penetrate plant roots, exchange nutrients and water for carbon. So it is a mutualism. They're both getting something out of it, the plants and the fungi. They're helping each other. And this mutualism really helps plants to acquire limiting resources, which you can imagine is really important under normal conditions, and even more so under things like the droughts that we've been experiencing in California. So really providing services to our plants that we care about above ground. So these services that organisms are providing are influenced by a number of things. Okay, so now we know a little bit about why we care. And all of these critical services, nutrient cycling, carbon sequestration, regulating plant community dynamics, what is that influenced by? And I want to unpack that just a little bit. The services provided by a soil community rely on at least 3 things. And I think they can be summarized into these 3 things. It's the amount and kinds of organisms present. So if we just think about microorganisms, bacteria and fungi and archaea, protozoa, they are everywhere. Bacteria are everywhere, but not the same kinds of bacteria are everywhere. And different bacteria, just taking that example from start to finish, different kinds of bacteria do different things. Okay, so yes, bacteria are everywhere, not the same bacteria are everywhere, and different bacteria can do different things. So the amounts and kinds of organisms present can influence the functions and the services provided by a soil community. The activity level of each individual organism also can influence the services and functions provided by a soil community. So what I mean there is that the below ground world, the organisms there have lots of different adaptations that allow them to survive stressful conditions just the same way, or when conditions and resources are scarce. So think about a bear hibernating in the winter. Why do they do that? They do that because conditions are unfavorable, and so they sort of shut down their metabolism. They slow their metabolism. They kind of go into this state that allows them to conserve energy and get through that harsh period until the conditions become more favorable. Now, organisms below ground have similar kinds of adaptations. They can go into dormancy, and actually a vast majority of microorganisms in the soil are dormant at any given period of time, just waiting for conditions to become favorable for them to turn on and start jamming again. So that the activity level of even each individual is also going to influence the service and functions provided by a soil community. And all of that is related to the physical, chemical, and and biological environment. But what I mean by that is interactions among organisms, competition, facilitation, mutualisms, all of those interactions influence ultimately what's happening below ground. And in terms of sort of activity levels and who is where, I think it's important for us to understand what influence, like what do organisms below ground require to survive, and it varies. So the adaptations and the ways that organisms below ground have adapted to make a living, to get by, is incredible. I mean, you hear about organisms, after Chernobyl, who evolve abilities to utilize these radioactive materials as their energy source, right? So organisms can get by in these just incredible ways, but they are all searching for the same thing. And organisms are searching for energy. And when we say energy, it's electrons. So we need electrons for all of our biochemical reactions. And you can get electrons different ways. We're also looking for carbon and nutrients. So the electrons help us give us energy, give organisms energy to make new cells, to grow and reproduce. But to do that, we need the building blocks of life. So we need carbon and we need nutrients. And all organisms below ground are searching for ways to get those two things. The majority of organisms in the soil get this by consuming organic matter, including plants, plant materials, and other organisms in the soil. And we can kind of like broaden that and simplify it to say there are 3 basic requirements for organisms below ground generally. And it's food. It's very similar to what we need: food, air, and water. Now, like I said, there are some organisms who are prefer maybe situations with no air. They actually do better when there's no air. But by and large, let's just sort of generally say that all organisms need food, or most organisms in the soil need food, air, and water. And when I say food, that's that search for energy, carbon, nutrients. Air is having oxygen, right? They don't, organisms like microbes don't breathe in the way that we do, but they need oxygen for respiration, cellular respiration. And then water, most organisms live within water films, for instance, in the soil. So water is a really important sort of resource for organisms, both biochemically, but also as a habitat, as part of their habitat. And this illustration below is from the microbe guide. It's describing different components of the soil. So on the left-hand side is sand, or different soil particles. It's a characterization of it: sand, silt, and clay. Sand is the biggest. These are soil particles that make up sort of that physical component of the soil. So sand is the largest, and it's that tan sort of circular space. The dark brown, medium-sized dot is silt. And then clay is this really tiny, teeny brown dot. And that's meant to represent different size characteristics. So sand, silt, and clay are different soil particles. They have very different sizes. But when we pick up a handful of soil, that, along with organic material, is often what we're sort of seeing and thinking about as soil, right? And if you move to this middle diagram, this pie chart, sand, silt, and clay, this is saying, okay, of a soil space, what is it made up of? And soil is about 45 in a sort of healthy, characteristic soil. 45% of it's made up of those soil minerals, sand, silt, and clay. About 5% is soil organic matter, this dark brown in this case, in this pie chart. But then that leaves another half of the below ground habitat as open space, pore space, right? So nothing. And that's actually critical, because you can think about the soil particles as the walls of the habitat below ground. Like, imagine it's a house. the soil particles are the walls, and it's really in between those walls that organisms live. Just the same way as we don't live in the walls of our homes, organisms below ground don't live in the walls of their homes either. And so it's that in-between space that's really, really important for the life below ground. And in an ideal scenario, the in-between spaces are made up of about 50% water and 50% air, so that you get that the organisms below ground get a little bit of both what they need. And if you compact a soil, what you're doing is you're scrunching up, you're squeezing, you're closing in the walls. And so that organisms have less and less and less space to live. And you can imagine that changes how this pie chart looks. And when you get something like a strong rain, there's now less pore space. It can fill up really quickly. And if your pores are full of water, there can't be full of air. So you start to lose some of the resources that these organisms need below ground. So this is why something like soil structure and soil compaction is really important for the life below ground. And then this right-hand side is just sort of rearranging that pie chart to show what it might, to sort of illustrate what that might look like in the soil itself. So looking at sort of the tan is soil particles, the blues are these in between like pore spaces filled with water and air, and then the black and the red are roots, organic matter, and fungi. Okay, so organisms, they below ground, they have the same basic requirements. They need food, air, water, but soil organisms, despite that, have diverse ecological preferences and optimal conditions. So just like we all need the same basic requirements, we need food, water, air, we might prefer different foods. I don't love the cold. You might love the cold, right? Some folks love to live in the deserts. Some folks want to live in the mountains. So we have diverse preferences and ecological and conditions that we can thrive in. And that's exactly the same below ground. So just as an example, just like birds, some birds may, live in grassland habitats. Some may live in shrublands. Goats may like to, may do better eating coyote brush, whereas cows are, will prefer the grasses, the forages. And trees can grow in different scenarios, right? So you have willows who like really watery conditions or moist conditions, and then you have blue oaks that can do well in upland conditions that are a little bit drier. So lots of preferences we're used to seeing and conceptualizing both among ourselves and among wildlife and livestock. And I just want to sort of take that understanding and apply it below ground and say organisms below ground have the exact same thing. Some may like it wetter or drier. Some may do well in warmer or cooler conditions. And when you pair that with really, really, really diverse conditions in the soil, so different amounts of resources, different climates, different substrates like parent materials, rocks, right? You just start to you layer all of that on top of one another, and you get all of these different habitats. And when you have diverse ecological preferences in the community below ground, that's when you start to just get these astounding numbers of diversity that are hard to sort of characterize in our minds, but is real. It helps promote that below ground biodiversity, is what I'm trying to say. So, okay, so that's a little bit about, well, hopefully why we care and why I would argue everybody does care, even if they don't know it, about the life below ground. A little bit about what promotes life below ground. And now let's unpack and just highlight a few of the many organ groups. So this is a diagram showing lots of different organisms categorized into different groups: microfauna, mesofauna, macrofauna, megafauna, based on their body size. This is one way that soil scientists sort of, many different ways to group organisms. This is one way to do it, is based on their body size. And we're going to just look at a couple of these groups I've highlighted and circled in the red. And we'll walk through them and talk about why they're important, a little bit about what we know about them in California, specifically on rangelands, and And we'll start with earthworms. So earthworms, they are really, I mean, this is one that I think many people, this is probably what most people are familiar with among all of these groups are earthworms, right? They enhance soil fertility by incorporating plant material into the soil. They help to redistribute it, take organic matter from the surface of the soil and bring it down, incorporate it into the soil. When they do that, they're burrowing holes and they're aerating the soil, bringing in that oxygen in those pore spaces that are really important for organisms like we just talked about, right? They can help bring water into the soil by increasing water infiltration. So if you have now these nice burrows, water can get through. Now you have, you're enhancing hopefully soil structure so that you can have air and water coming into the soil, which again is important for most organisms living below ground. So they're really helping to be these engineers of the soil. We know of about 7,000 species globally of earthworms, and they're often grouped sort of ecologically based on where they live in the soil and what they eat. So there's 3 kinds of earthworms that we highlight in our microbe guide, and it's epigeic earthworms, endogic, and anisic. And this is based on, again, what they eat and where they live. And so these epigeic earthworms are the ones in the yellow here. They live sort of at the surface, they're eating litter. And a lot of the earthworms that do this are sort of what we would consider like earthworms you'd find in like a compost bin. Then you have endogic earthworms that go a little bit deeper into the soil. They're eating plant litter at the surface, but also consuming just organic matter within the soil profile and helping to cycle those nutrients that way. And then anisic earthworms take soil organic matter from the surface and they make these like permanent, really deep burrows. And so they actually influence the soil in unique ways. You can imagine when an earthworm is just hanging out at the surface or going a little bit deeper down or making permanent burrows, that might influence the functioning differently, right? So again, this is that idea that not all organisms are the same, even if they're the same kind of organism. So earthworms, even within earthworms, can influence the soil in different ways depending on who they are and what they do. So these are a couple of examples of earthworms that are found in California. The yellow is an epigeic earthworm. It's a non-native redworm. The blue, the endogic, you have an example highlighted in blue of the 2 kinds of earthworms that we show in the guide. One is a non-native gray worm, and then the other one, that blue and red one, is actually a native species of earthworm. And then one of the most sort of iconic anisic earthworms is Lumbricus terrestrius, which is the night crawler, that folks use for fishing. So it's actually pretty invasive in a lot of places. In California, we do have both non-native and native earthworms. That's not the case everywhere. So in the Midwest, where I'm from, glaciation pushed down, all of the topsoil, and with it, we lost all of our native earthworms. So the earthworms in the Midwest are invasive and really changing the ecology of these systems, helping, for instance, in some cases, invasive plants to take over our woodlands. So they can function differently. Natives tend to dominate in uncultivated areas, grasslands, non-native and cultivated areas. So that would be, our croplands. And densities can range between 1 and 4 per liter. So nematodes are a different kind of worm. They're round worms. And I think most often folks think about nematodes as being parasitic to plants. And for me as a soil ecologist, what I think first of when I think of nematodes is actually nutrient cycling. So nematodes eat bacteria, they eat fungi, they can eat protozoa or even other nematodes. And when they do that, I'll say it very scientifically. So let's say they're eating bacteria. When they eat that bacteria, they spill some of the bacterial guts into the soil and it releases nutrients. That's the way I think about it. Well, like to describe it at least. So when they're eating microorganisms, in that process, they're releasing their guts and nutrients. Okay, and that's making it available to plants. So they're taking nutrients that were previously unavailable because they were bound up in microbes, and then they're releasing it back into the soil system for plants to take up. So they enhance soil fertility. They are the most abundant animal on Earth, and we have described only a very small amount of species that we think exist. They live in water films. So this is, again, why water is so important within the soil, one of the reasons. And they live in these water films surrounding soil particles and are really sensitive to water availability. But when conditions are harsh, you know, just like many other organisms in the soil, they can enter this reversible dormant state. And they are characterized in taxonomically, you can characterize them, but sort of if we want to think about their ecology, they're grouped into different sort of feeding behaviors. So you can have, and these are illustrations of nematode feeding mouthparts. And you have omnivorous nematodes that can eat on lots of different things. You have plant feeding nematodes. So these have feeding mouths, essentially, or parts that allow them to feed off of plants. And then you have bacterial feeding nematodes that look differently and have different structural features as well. In the Sacramento Valley, I just want to give an example of sort of the amount of nematodes we know can exist within soils in California. There's between 1,000 and 4,000 individuals of nematodes in a liter of soil. And some, like, just a cool little factoid on the Central Coast, there's a type of nematode it's entomopathogenic, so it kills insects, and it suppresses lupine ghost moths. And in so doing, it reduces the damage of these moths that are caused to lupine plants. So I actually have this beneficial indirect interaction with lupine plants via infection of these ghost moths. Okay, so protozoa, so similar to nematodes, when I think about protozoa, I think about nutrient cycling. Because protozoa eat bacteria, and they can eat other protozoa, they can even eat fungi, although we often think of them as eating bacteria. When they do so, that they release that nitrogen into the soil the same way they're releasing the guts of the bacteria and making that nitrogen available again, enhancing soil fertility through their predation. There's about 1,600 known species of protozoa globally. They're super diverse taxonomically, but they're generally characterized into 3 general groups based on their body type and how they move. And so these are ciliates, and this is another, just amazing illustration by Mel. So on the top is an example of a ciliate. So they have little hairs and they wiggle about. In the soil, you have flagellates, and they have these flagellum that allow them to move by whipping, sort of these whip-like structures that propel them forward in the soil. Again, in these water films surrounding the soil particles. And then you have 2 different kinds of amoeba. You have testate amoeba and you have naked amoeba. And the testate amoeba have this hard shell on them that allow them to be pretty resistant to dry conditions. But it comes at a cost to moving around and really being able to move for resources. The naked amoeba can kind of ooze around the soil a little bit more, so it's more mobile. But they live in these water films and they can eat bacteria, but also many other things. And again, really enhance soil fertility. Just another example to give us a sort of sense of what we might expect to find in California. So cultivated grasslands, previously cultivated grasslands, so have been tilled. Some studies have shown up to 400,000 protozoa in a tablespoon of soil. And that community in that example was dominated by these flagellates. Rangelands on the Central Coast, we've found an average of 5 million protozoa in a tablespoon of soil, with the community dominated by amoeba. And there are some generalizations that folks try to make, with fungal dominated soils tending to have more testate amoeba and bacterial dominated soils tending to have more flagellates. So thinking about them as indicators of, the community at large, there are some generalizations that that can be made, but there are limitations there. Okay, so fungi, we're moving on to fungi. So this is probably the other group that folks think most about. We see mushrooms on our landscapes all the time. Fungi influence are really, really important. They influence forage productivity, plant composition, all of those things that we were talking about at the beginning. Some of them form direct associations with plant roots, and some of them just decompose organic material and recycle organic material. There's about 97,000 known species of fungi globally. Many of them, the mycorrhizal fungi link with plant roots. So again, I mentioned this at the beginning, but in that mutualistic interaction, the fungi provide limiting resources, so phosphorus, nitrogen, and water to the plants in exchange for carbon. So it's this mutually beneficial relationship. And it can be really, really important for plants. It helps plants grow. It helps them survive and thrive normally, and especially under stressful conditions. So really critical. Most plants associate with some kind of mycorrhizal fungi. There are different kinds of mycorrhizal fungi that's illustrated in this other beautiful illustration by Mel. Arbuscular mycorrhizal fungi, which are shown sort of represented by the orange in this illustration, associate with the vast majority of like herbaceous plants, so grasses, forbs, that kind of thing. They can associate with some trees as well. Ectomycorrhizal fungi form associations with a much more narrow group of plants, things like oaks and willows, Doug firs, that kind of thing, aspens. Aspens, I believe. And so associate with different kinds of plants, but performing generally the same functions. And then you have saprotrophic fungi that don't form direct association with plant roots, but they help to decompose plant material and recycle nutrients within the system. Fungi are thought to be really important in building soil organic matter and sequestering carbon. And I think that they're talked about a lot more than bacteria in this case, or other organisms. And I thought maybe I'd just spend one slide thinking through why that might be and talking through why that might be. It's not to say other organisms aren't important for sequestering carbon and building soil organic matter, but mycorrhizal fungi, they do help plants to acquire more water and nutrients through those mutualistic associations, and thereby capturing more CO2, so promoting photosynthesis and ultimately improving production. And we know that a lot of that carbon is going to go into the soil to build soil organic matter and sequester carbon. So by helping plants grow, fungi have this like really unique role in sequestering carbon. There is some evidence that like these decomposing fungi, these saprotrophic fungi, may eat the carbon that enters like the organic matter of the plant litter more efficiently than bacteria. Now, this is sort of a gross generalization, but it's something to think about, and is how efficiently organisms recycle those nutrients into the soil. Do they keep a lot of it in their bodies within the soil and byproducts of their metabolism in the soil, or are they releasing a lot of it back out as carbon dioxide in the atmosphere? We know that this is really, really important for carbon sequestration and building up organic matter. And there is some evidence that saprotrophic fungi can eat that pretty efficiently and keep it in the soil. And then fungi have a really important role in enhancing that soil structure that we're talking about in aggregation and making a really nice home below ground. They produce glue-like substances which can protect soil carbon and make sure that that soil is habitable for all of the other organisms that call it home as well. So really important component of the system. I'll just wanted to give a nod to the sort of fungi to bacteria ratio. So this is a popular proxy for fungal dominance. So fungi are really important. There's I think many people hear discussions around fungi to bacteria ratios. And from and a lot of magic numbers will be thrown out there. And I'll say from my perspective, I'm not not sold on there being one magic number, like a 5-to-1 bacteria, fungi to bacteria ratio, or 10-to-1, anything like that. I think, values reported and from my work and others across California rangelands, the ratios are going to vary by site and season, and they range from 0.01, so largely being bacterial dominated, to 5, which would be 5-to-1 fungi to bacteria. So lots of variations that can happen just across space and across time. Ecological interpretation of fungi, of sort of these ratios, can be tricky when we're thinking about cause or effects. Are they responding, or what is it driving? But more importantly, I think that what I would encourage folks to do if they're trying to look at and think about and use indicators of, you're taking soil samples and you're sending it off to ward labs and you're looking at the microbial community, I think that's awesome. And what I would say is, encourage you to do is to think more about total biomass of both fungi and bacteria, because you can have any given ratio, and not a lot of them, right? You can have a 10-to-1 fungi to bacteria ratio, but you don't have a lot of fungi or bacteria in your soil. So what I think is more important is probably increasing the biomass and abundance of all organisms. And so looking at that total biomass or abundance number can be really important. And I hope that folks will start to think about that as well. The final group that I want to sort of highlight are bacteria. So bacteria, I don't know. I'm a bacteria girl. I love bacteria. That's what I studied for my PhD. I studied nitrogen cycling. I think that sometimes in the soil health world, they don't get as much attention as they ought to. Maybe that's okay. Maybe that's maybe that's warranted. But I would like to sort of just take a moment and highlight all the important things that bacteria do as well. There's about 15,000 described species, but we think there's many, many more. There's thousands of unique kinds of bacteria in a handful of soil, more than there are plant species across California. And one notable group of bacteria is nitrogen fixing bacteria. So along with some archaea, they're the only organisms on the planet, except for humans now with the Haber-Bosch process that we make fertilizer with. They're the only organisms that can naturally make atmospheric nitrogen, which is atmospheric nitrogen available for plants to use. And the atmospheric nitrogen is 78% in the atmosphere, but it's unavailable for plants. And so these organisms can take that critical resource and make it available for plants in a way that no other organism, not fungi, can do. So super, super important. Soil bacteria also consume methane. They're methanotrophs. So in the soil, organisms, these bacteria can consume methane. Having a healthy microbial community won't get us to a point where soils consume more methane, for instance, than livestock produce, but having a healthy soil can help to minimize growth of atmospheric methane concentration. So soil bacteria doing that really critical process of consuming methane as well. It's just one other example of the importance of soil bacteria. Here are some other notable biota. So we have arthropods, which are, dung beetles, like we talked about, spiders, centipedes. Archaea. Now, archaea don't play a big role in rangeland soils, but are responsible for methane production and rumen of livestock. So that's one of the areas that I think many of us would be familiar with them. They're very similar to like bacteria. They're single-celled microorganisms. Biocrusts are super important, so especially in semi-arid rangelands. So these are really like assemblages of different kinds of organisms. You have cyanobacteria, algae, mosses, lichens, bacteria that fix carbon and protect soil. So really, really important in many arid, semi-arid rangeland systems are biocrusts. Phytophthora, water molds. They're not fungi, but in California, they cause sudden oak death. And here's another figure from the microbe guide of a tan oak twig that is showing symptoms of being infected by Phytophthora. So a water mold, that's really, really important parts of these systems as well. And then viruses. So viruses are really cool and are a part of the system that more and more and more we're learning about as scientists. They influence, so remember how I talked about protozoa and nematodes influencing nutrient cycling by feeding on bacteria and other organisms. Well, viruses act in a very similar way by infecting bacteria and other organisms and lysing their cells. We know a lot about viruses and nutrient cycling in water, like in aquatic systems, and we're learning more and more about them in soil systems. They can affect plant community dynamics. So folks at UC Davis have done some work looking at barley and yellow dwarf viruses. They're transmitted by aphid insects and affect native grasses, and can influence like competition among plants within our rangeland system. So super cool. I think there's a lot for us to learn there related to viruses. And so then we end here, and I'm so sorry, and I think I'm going to have to, and I hate to do this, I think we're going to have to just either pause for a second or take you with me. I had a major oversight and did not plug my computer in, and I am getting warnings that my computer is going to die. So if everybody can just sit tight for 1 second, I'm sorry to do this. I'm going to run and get my computer plugged so I don't lose you all.
The organic part of a webinar.
Yes, I'm so sorry. Just give me 1 second, everyone. Stay tuned. Thank you.
And this actually, as we're all here today, just if anyone has, we'll have Q&A in just a couple of minutes. So anyone who's interested in adding anything to the Q&A, this is a great time to do it. I see that people have already asked questions specifically about some of the organisms, whether that's about the dung beetle, and then Lawrence asked about how healthy soil, right? How if soils are composed of abundant and diverse organisms, how we can be planting in essentially dead soil with chemical fertilizer. So a couple of good questions, but this is a great opportunity for us to add any questions if you.
Awesome. Thank you for that impromptu break. Okay, I'm plugged in now, and we can wrap up. Can you still see my screen okay? It's all okay?
I think so.
Okay, perfect. All right, so let's take this home. So now we've just talked through some of the groups that are really, I don't know, fun to think about and important components of the system. And we've had a moment to think through why they're important, what they're doing below ground, both for us and just for the ecology of our landscapes in general. And so we have an opportunity to steward the below ground world. That's how I like to think about it, is that we're stewards of life below ground. How can we steward soil health? How can we steward the organisms that rely on our stewardship and that we rely on so heavily for all of the services that they provide? And the place that sort of my mind goes back to is, well, in order to know how to steward them, we need to know what their preferences are, what they require. And that's why I started with that at the beginning. They require food, air, water. We know, that most of the food that they get is from plant material or other organisms. We know that water and air can be affected by soil compaction and the structure of the soil. And so we can take that information and we can translate that to begin to understand what we can do to steward organisms below ground. And luckily we have some soil health principles already that I think are fairly well known, becoming well known. And I've put them up here and added one for myself as well. And these are all based on that understanding. So the first is maximize soil cover. So we can if organisms they need organic matter and good soil structure to have food, air, and water. Maximizing soil cover, whether it's through living plants or residues that are left on the ground, not having bare ground, that can really help make sure that the habitat that's below ground for organisms is quality habitat, and that they are getting the food that they need through decomposition of organic matter. Promoting living roots is another one. So a large amount of food that organisms get below ground come through roots, actually. not just through the roots themselves, but through, and I'm sure some folks have heard either the liquid carbon pathway, but root exudates. So essentially sugars and other simple compounds that plants push out through their roots help to feed the below ground world. So promoting living roots, both through space and across time. So extending the growing season, perhaps filling in spaces within the landscape spatially, can help to promote life below ground. Fostering biodiversity above ground can foster biodiversity below ground. So biodiversity above ground, one of the most obvious is diversity in plant communities, right? So whether you're grazing to promote grassland biodiversity within your forages, or you're seeding in diverse mixtures, or embracing diversity of habitat types within your property boundaries. So that could be, oh, I have a little bit of forest, I have some grassland, I have some shrubland. That diversity at that landscape scale helps to foster diversity below ground as well at that landscape scale, because remember, again, organisms have these different preferences, and we'll find different ones in forests than we do in grasslands, etc. And then foster biodiversity can also mean sort of having things like stacked agriculture can be one way to foster and think about biodiversity as well. Minimizing disturbance, so compaction and on the flip side, tilling. Again, because this structure of the soil, the house, like when you compact soil or you till it, you are damaging the structures of the home, right? You think about the walls of your house, you are either squeezing them in or you are ripping them apart, and you're not, and that's what you're doing below ground for these organisms. So minimizing that disturbance so that they have really solid homes with pore space in between that air and water can flow through, and that they can get their food within and move around in. And then the other one, so those are principles of soil health, and then reducing use of biocides is another one I want to add. Things like dewormers, even, and herbicides, pesticides that can sort of cascade through the system and have influences below ground that are unintended. So just thinking about careful use of biocides and thinking through the implications of their use from a below ground perspective will be important. And and this is my final slide, just giving a nod to the fact that healthy soils, and I haven't defined soil health, but healthy soils, just the way that sort of healthy humans are, it doesn't always look the same. Okay, there can be different characteristics. So this is a serpentine grassland on the left-hand side and a non-serpentine grassland on the right-hand side, different environments. But serpentine grasslands have really unique soil characteristics that actually promote biodiversity of, and refugia for some of our rarest native plants now. They're like very special habitats. And a lot of that comes from this unique aspect of the soil. So what a healthy soil, serpentine soil looks like is going to be different than other soil types. This is just one example. So they're not always going to look the same, but invariably soil organisms are going to play a key role in maintaining the health, whatever it looks like, and helping to achieve goals, whatever they may be. And so I'm going to end with that and put my email up here and say thank you so much for your attention, for your patience as I had to go get my charger, and welcome any questions.
Thank you so much, Dr. Carey. We really appreciate you taking the time this has been a very thorough presentation. We got a number of questions, but I think for those of you who continue to have questions, we will be sending an answer to one of the questions was whether we would be sending a recording of this webinar, and we will be in the next couple of days. So expect an email with this. If you are whether you're registered or attending this full session, you will be receiving a follow-up email, and this will have all of the links attached, and including Chelsea's or Dr. Carey's email. First, so a couple of questions. We started with ones. Lane was asking, can you comment on depth of soil? Just their actual question is how deep is soil, but I know that varies so much. So how would you answer that question?
Yeah. Yeah, it does vary a lot. I mean, soil can go, I mean, we're talking meters deep in places. So it's commonly we'll dig soil pits to a meter, 2 meters, but it really does depend. It depends on how old or young the soil is. It depends on if it's an eroding landscape. Um, and if you're sort of on an eroding landscape, for instance, you might have a lot shallower soil. When you're nearing, when you're hiking up in the mountains, your soil is going to be a lot shallower in places than maybe if you're in a depositional area like in the Central Valley with these deep soils, or thinking about in the Midwest, these soils of these mollisol, these like prairie soils. So they can go meters deep. And then in some places, you're looking at a lot shallower. So it just depends. Yeah.
Thank you. And Larry asked could you comment on the aging of soils? Do bioconditions just change, or can they become permanently inert?
Okay. So the characteristics of soils are influenced by a number of soil forming factors, we call them, that Hans Jenny and sort of really solidified for us as a scientific community. And the acronym is CLORPT. So it's what makes a soil, and it's the climate, the organisms, the relief or topography, the parent material, and the time. And so time is a really important aspect of soils. And there are certain things that happen as soils become older, as they age. A lot of you'll get more like iron and aluminum oxides. You have a lot of weathering, it's called. So you'll have transformation from like primary to secondary minerals, and they often will become finer over time, soils. And so that does influence, and they can, and in weathering, they can have other influences as well, like the changes inherently in pH and things like this. And so there are definitely real shifts that happen with time. They don't become inert or dead or lifeless, but there are characteristics that will change as a soil becomes older. You start to get a lot more horizonation, we call it. So when a soil is young, it doesn't have a lot of differing characteristics throughout the soil profile. You might dig a pit and it just kind of all looks the same. Like it's all just one color, it's all the same texture. But if you give it, tens of thousands of years, it starts to have really distinct layers that are very characteristic of the organisms that have been there, what the rocks that formed the soil, and have a lot more a lot more detail as you look through it in the soil profile.
You visualize that from the beach, right, where you can see those years just on the cliffside there.
Yeah.
Can you just speak a little to how you identify the microorganisms in the soil?
Yeah, totally. Yeah. So there's lots of different ways to identify organisms in the soil, and I'll talk about the microscopic, but I mean, there's lots of organisms that you can see in the soil just by getting a shovel and digging up some of the soil yourself and looking. Do you see earthworms? Do you see evidence of earthworms? Do you see other life in the soil, right? And then when you get to these microscopic organisms, like microorganisms, we have sort of the bread and butter techniques these days are called DNA-based technologies, so molecular technologies. And so it's either DNA or RNA based. And what we can do, so what I often do in my work is I'll take a sample of soil and you isolate like a particular gene. And that gene will tell you is found like in all bacteria, for instance, and it tells you though a little bit about their taxonomy and their phylogeny, how they're related. So I can take a sample of soil, I'll do some preparations on it, I'll send it to what we call a sequencer, and it sequences in this case, it would be like a 16S gene. It's a particular gene for a bacteria. All of them have it, but it varies. And so then now, based on information on that gene that I found across that soil sample, I can compare it to a database of known genes of bacteria, and I can start to understand who is in my soil. You can do same thing for fungi, for all sorts of organisms. You can extract their DNA and look at them taxonomically. There's other ways to do it where you sample all of the genes. So we have genes that tell, like bacteria have genes that tell you who they are, but they have genes for doing all of the functions that I talked about, nitrogen cycling, carbon cycling. And you could take a sample and you could say, I want to sequence all of the genes. And that's called shotgun metagenomics. And that tells you a little bit more about the potential function of your soil as well, based on the genetics of the organisms who are in there. So lots of like really cool new ways that we can start to look at who's in the soil and what they're doing. I mean, there's even more. That's just like two that I'm happy to talk more about, but it's super cool. This is the last one I'll say, is it's called DNA stable isotope probing. And you take different elements have different amounts of neutrons, like they weigh different amounts. And so you can label plant material. You can grow plant material where it takes up carbon dioxide that's heavy. And now it's like labeled. And now when that moves through the soil community, whoever eats that plant material that's heavy, they become heavy. And now we can separate the heavy microbes from the light microbes, and we can sequence them using those DNA technologies and see who was eating the plant material and who wasn't in my community. So super cool techniques that people can do these days to get at who's there and what are they doing.
Oh, thank you. That was such a thorough, I mean, and it sounds like just scratching the tip, but it's also really exciting to hear your passion for this, right? I mean, just opening up that world, how cool that is. Okay, will you redefine forage productivity? Someone was asking about that.
Will I define forage productivity?
Yeah.
It's how much forage you grow. So it's just biomass. And in California, in our annual system, we often measure it at peak biomass, right? So depending on where you're at, sometime between April and June, when you have the peak amount of forage, that's typically sort of the amount that you'll grow in the season. And we can measure it by like clipping the above ground, sort of a portion of forage above ground and weighing it. Right. So in these annual systems, that's how we can get at above ground forage biomass, but it's just how much forage you grow in a given year.
Okay.
Yeah.
Let's see. I think, this person asked about the best soil for plants. I grow at home, like tomatoes and potatoes. They're sort of asking specifically. You talked about that in terms of how to care for it, but would you add on to that in any way?
Yeah, I would point folks to, I would say even like local, because different plants like different soil conditions. And I would point you to like your local, master gardeners group or something to understand for particular like horticulture types and endeavors and plants, like what conditions they prefer, because it's going to vary. Yeah.
Mm-hmm. Oh gosh, all these at the end. Okay. But that was a question that this Lawrence was sharing about. There, healthy soils, which are composed of abundance and diverse organisms, are so important. Why are food plants so healthy and productive when grown in what he said, dead soil with chemical fertilizers and that, nitrogen, potassium?
Yeah. How do I say it? So in dead soil where we dump chemical fertilizers, plants can grow abundant and have, high nutrient density, because they are getting what they need, but not in a sustainable, like natural way. If that, for a better, lack of a better way to say it. So the soil community does that naturally, And if we kill it, we have to supplement. And that's what we've done is we've created a system where if our soils are like dead or not providing the services that we want, they don't have a lot of organic matter, there's not a ton of organisms in it. Well, now what do? We have to now we have to step in and add chemicals. And so yes, it has done what we've needed it to do, but it has come with a lot of consequences. It's come with a lot of and those chemical fertilizers further sort of destroy the soil system. And they have obvious, like so many other environmental repercussions, from air quality to water quality, energy production, all of that stuff. And so it's really just not a sustainable way for us to continue moving forward. And so if we can re-embrace soil health and the organisms who are there and naturally want to and able to do this for us in a healthy way, how can we sort of shift our mindset and start to rebuild that natural capacity of these systems to do that, so we can reduce our reliance on these chemical inputs?
Yeah. Okay, I want to value your time. We have a number of other questions, and we'll talk afterwards about how we want to field questions, because boy, there are a lot of wonderful questions that people are asking. And I know a couple of you are asking for specific information on the research being done at TomKat. And some of these things we can give you some good links on to set you up with some good information that you can read about, because there is incredible work that's being done at TomKat Ranch. But for now, we're gonna put a pause in that, and we're gonna free people, although I know we're on the webinar, so you all could have taken a potty break during the time if you needed to, but wanted to say thank you again, Dr. Carey, so much for taking this time, for being here amidst a very, very busy schedule to share this information and get people really psyched on some of you who have a background knowledge, but for a number of us that have learned so much in this presentation. So thank you.
Thank you. Thank you so much for the invitation. It was my pleasure. Thanks everybody for spending a Saturday morning with me. And yeah, reach out if we'll try to follow up on all the questions, and reach out if folks have any other things that they want to talk about, or if you want some guides in print or anything like that.
Thank you. Thank you. And for those of you who are here, just remember you will be getting a follow-up email in the next couple of days. So take it, spread it around, just get share this information, and you know how critical it is. And the more we know, the more we are geared into the decisions we're making at home, and the decisions we're making environmentally, and when we're making decisions voting wise, and when we're donating and all of that. So we'll give you links to check out more about TomKat Ranch and Point Blue Conservation Science. Also, if this is a time that you're able to donate, please remember we are run by donation. So both this webinar on the Junior Land Stewards Program, which we're going to be also picking Dr. Carey's brain on for moving forward with the Junior Land Stewards Program, all of these things. And then of course the important, the service, the stewardship and conservation of this shared open space land, critical. So please be thoughtful of that during now or your time of donation. And keep us up. We'll keep you up to date on all social media. So things coming up. We've got some work days coming up. We've got Pescadero, the Old Town Purísima site. We'll be doing a couple more tours there. We've got a couple of exciting webinars coming up too. So just keep your eyes open. Just open up those links when you get the emails. We try to keep them pretty sporadic, but we're trying not to send you too many, but please make sure that you open those up and see what's coming around, and join us when you can. All right. Thank you again, and have a wonderful weekend.
Thank you.
Bye, everybody.